Assalamualaikum. My name is Nishika binti Muhammad Khairi Sham S50065. Today I will present a review on the effect of microplastic on oyster. So, introduction, what is filter feeder organism? Filter feeder organism is an aquatic animal that feed on particle or small organism strained out of water by circulating them through its system. This organism continues to clean our water will decrease the turbidity and ammonia of water. They feed by pumping water through their gills, trapping particle of food as well as nutrient suspended sediment and chemical contaminant. So this is an example of filter feeder organism, which is oyster, mussel, and clams. So what is oyster? Oyster are belong to the family Ostreidae, class bivalve in the phylum Mollusca that live in the brackish and marine water. The body of oyster has two calcareous whales that join together by a ligament along the narrow hinge line. The alteration of water quality may affect the oyster. Consequently, the loss of oyster reef and other bivalves, thus disrupting the nursery habitat of crustaceans and fishes within the estuary. Oyster attach to the substrate and require hard bottom areas. So what is microplastic? Microplastic is plastic debris can come in all shapes and sizes, but those that are less fit than 50 millimeter in length are called microplastic. Pollution of microplastic particles are derived from the breakdown of larger debris that enter the environment as microscopic fragments. Because filter feeding organisms ingest microplastic while feeding, they are likely to be impacted by microplastic pollution. These chemical substances that detected in microplastic become toxic to living organisms when subject to high concentration. So my problem statement is filtering of water containing microplastic will detrimentally affect the ingestion rate of oyster. The reduced ability of oyster to ingest food will lead to the lack of nutrient in the oyster. This will eventually affect the food chain, especially in the area with high dominance of oyster. So my significance of study. This review study also can be used as a reference for marine policy management. The awareness of Malaysia can be built stronger from the action to control these big issues can be held better. Provide information to ensure the safety condition of environmental and human health. This review study used as a benchmark in the determining the level of water pollution. So my objective is to compare the filtration rate of oyster at different microplastic densities, to determine the finding of the sediment stability on the filtration rate of oyster at different density of microplastic. So methodology first is search strategy. Search article from database namely Google Scholar, Scopus, ScienceDirect, ResearchGate, Elsevier, Sprinkling, and Semantic Scholar. Use keywords such as oyster, filter feeder, microplastic, sediment, filtration rate, and ingestion rate. So next is data collection. Article will choose by screen the title, abstract, and to the full article from the database research. Only papers that published in English were accepted. And lastly, data organization and reporting. Information seized from each study were organized particularly according to the data about filter feeding organism, filtration rate and sedimentation, experimental method result, author name and year of publication. So this is my result and discussion. Filtration rate of oyster in different concentration of microplastic. Low concentration with 0.25 to 0.80 filter 70% of algal cell clear. Then for control treatment, oyster will filter 80% of algal cell. High concentration with 25 to 80 of microplastic, then only 50 of filtration rate in percent of algal cell clear. Based on the graph, it shows the high concentration rate of microplastic. And this is my result and discussion. Filtration rate of oyster larvae were decreased in high concentration because of environmental factor and the microplastic clog up in the stomach. Filtering of indigestible plastic particle can block nutrient absorption and cause damage to the digestive tract of animals. The larger particle of microplastic could probably be filtered by the oyster, but instead of being ingested, they are retained within the shell cavity by adhesion. Marketable bivalve further clarify the uptake and ejection dynamic of larger particle and the associated potential risk for consumer. So this is graph for sediment stability on the filtration rate of oyster at different concentration of microplastic. Low concentration with 0.25 to 0.80 filter 60% of algal cell. Then for control treatment, the filtration rate was 70% of algal cell clear. Lastly, for high concentration, oyster will filter 40% of alga cell. Control treatment has the most diverse of species and the highest number of individuals in a mesocosm compared with high and low densities. The sediment and the organic matter content such as mechanism, detritivore and plant origin to become their food source. Caused by the mortalities, reproduction rate decrease and less feed because of the filling fuel, and all of this can be originate from the blockage of microplastic in their digestive system after filtration. Organisms in the sediment can be affected by the impact of microplastic when deposit feeding it filter process or filters. Filtering of indigestible plastic particle can block nutrient absorption and cause damage to the digestive tract of animal, as well as the long foraging and shelter function of the habitat for associated reef fishes and crustaceans when sediment fill the interstitial space among the oyster shell. So my conclusion is the filtration rate of bivalve was getting lesser as the concentration of microplastic increased. This may be due to the digestive system of the oyster are clogged up with the microplastic particles. Microplastic also can cause several harmful physical effects on human and living organisms through such mechanisms as entanglement and ingestion. Government and also organizations take many intensive work project and management. Bans are becoming an increasingly popular way of curtailing their use, and limited evidence indicate that due reduce the risk. So these my references. That's all from me. Thank you.